Hello friends, I am Dr. Prashant Sharma and you are watching MEDICO'S Hub. In this video we are going to discuss the innate immunity under the heading Defense by Inflammation and Fever. What is inflammation? First we'll discuss the inflammation. It is the protective response given by our tissues against invasion by pathogens or antigens. Inflammation is characterized by few symptoms like rubor, dolor, calor, and tumor. Rubor means redness, then pain, heat, and the swelling. Now, how this inflammatory response mediate? It will occur in 3 steps. First one is vasodilatation and increased permeability of blood vessels. Now, how this works? This is skin or mucous membrane, and this is a blood vessel. The first step is that entry of microbes like bacteria occurs. Then it results in damage to tissues. As a result, tissues give inflammatory response. This inflammatory response is in the form of vasodilator. These vasodilators include histamine, bradykinin, prostaglandins, and complement proteins. Histamine is mainly contributed by mast cells, and from the blood vessels it is contributed by basophils and platelets. Bradykinin is contributed by tissue kininogen, while plasma kininogens also contribute to it. Damaged tissues contribute prostaglandins, and obviously the complement proteins are contributed by plasma, and all of these vasodilators will finally result in arteriolar dilatation. So first step is vasodilatation and increased permeability of blood vessels. Then comes the second step, that is phagocytic migration and phagocytosis. How this step is initiated? For example, this is a blood vessel and these are endothelial cells. So first the vasodilatation will occur and the vessel will become like this. First important change is that the endothelial cells will express adhesive molecules or adhesion molecules on their surface. Endothelial cells express adhesion molecules, which are basically proteins, on their surface. Now, as a result, the circulating blood vessels, they will start adhering to these adhesion molecules. Circulating WBC, especially phagocytes, attach to adhesion molecules. Then gradually they will come out of the blood vessel, and that is known as diapedesis. And finally, under influence of chemicals, they will move towards the pathogen or antigen and perform the phagocytosis there. It's important to note that first cell to come at the site of infection are neutrophils, followed by monocytes and macrophages. So this is the phagocytic migration and phagocytosis. We know that how the phagocytosis will be performed. The endosome is formed, phagocytosis is done, endosome is formed, it is merged with the lysosome and then finally digested and ingested. Now the third step is tissue repair. After these interactions, tissue repair will occur by migration of fibroblast that will form collagen fiber, then scar is formed and repairing is initiated. Epithelial cells, keratinocytes and macrophages. In the whole process, it's important to discuss about nuclear factor kappa light chain activator or enhancer of beta cell. What is this? This is basically important for transcription of genes to promote synthesis of chemicals that mediate inflammation. But this factor usually remains inactive because it binds with IKB alpha. This is a cytoplasmic protein which binds with NFKB and keeps it inactive. So many anti-inflammatory drugs actually promote the synthesis of IKB alpha so that NF-κB may remain inactive. Now the fever. There are three important steps of fever which help in or play an important role in immunity. That is, these inhibit the reproduction of pathogens. It enhances the synthesis of interferons and it helps in synthesis of enzymes that prevent the microorganisms to grow. So these are the various roles of inflammation and fever. In the next video, we'll discuss the complement system. Hit the like button, share and subscribe our channel to get the latest updates and notifications.